Hey everybody, welcome back to another lecture video. This on first of two on protein synthesis. This first one is transcription. So we're going to be taking the genetic information stored in DNA and actually writing that into a RNA template that can be used in translation or in the next lecture. Okay, so transcription controls the expression of the genes and the production of proteins. And some of the general features in RNA synthesis are that RNA initially synthesized from a DNA template. All four ribonucleoside triphosphates are required, as is magnesium 2+. We saw that the same during replication with DNA. In this case, a primer is not needed to start RNA synthesis, like we needed an RNA primer for DNA polymerase in the last chapter. But DNA template, of course, is required. And the RNA chain is going to grow in the same direction as the DNA chain did from 5' to 3'. It has the same basic structure, it's just missing the 2' hydroxyl group. Oh, I'm sorry, it contains the 2' hydroxyl group. That's the only difference. So as far as connecting them together, the polymerization is the same. The enzyme that we're going to look at, polymerase, uses one strand of the DNA as a template, and the base sequence of the DNA contains the signals for initiation and termination of that RNA synthesis. And we're going to investigate that a little further. The enzyme itself will have to move along the 3' to 5' strand in order to synthesize a 5' to 3' piece of RNA. RNA polymerase is the molecule that we're ultimately talking about here. Now, RNA polymerase is the enzyme that catalyzes this RNA synthesis. Its molecular weight about 470,000. I was going to say 4,700, but 470,000 just sounds funny. 470,000 Daltons. I believe that's just the molar weight given in the unit we usually use for proteins. It's a multi-subunit structure. There, I believe, is something like 6 different subunits. There's an alpha, an omega, a beta prime, a sigma. The core enzyme, so I'll give you some more info. Core enzyme is a couple of these alphas. Sorry. Alpha 2, the omega, beta prime. This is the core enzyme. It lacks the sigma subunit. The hollow enzyme, which is what it's called in its functional form, has all its components, including the sigma subunit. We'll see that sigma subunits are important here for the initiation of transcription. So you can see here the general parameters that we're going to be looking at. We're going to have a coding strand. This is what sometimes also called the sense strand. Let me clean this up. So coding strand is the sense strand, or sometimes the positive strand, or the non-template strand. This is the same as the sequence of the RNA that's going to be synthesized. So if you can see here, the coding strand has an A, T, G. So we would expect to see the same exact letters there. Of course, RNA has a U instead of a T. So same sequence of information. Now then there's the template strand. This is also referred to as the antisense strand, or the negative strand, or of course the template strand. And this is the one that the hollow enzyme is going to bind to and transcribe. Now the sigma subunit here, we'll talk about the sigma subunit in a minute, but let me jump back real quick and say, now this first process of RNA polymerase binding to the template strand and making this RNA is referred to as transcription. Now the next lecture we'll take that mRNA and actually encode it into a protein. Okay, the sigma subunit recognizes a region on the DNA called a promoter region. promoter region. Now, the sigma, after it recognizes the promoter, it starts transcription, and then after transcription starts, the sigma subunit will get released. Now, the promoter regions are portions of DNA to which the RNA polymerase will bind at the start of transcription. And you can see here some of the consensus sequences that we see for various different organisms. So these are are these various different organisms? Let me see. Actually, these are all E. coli. These are all promoters of E. coli. And so they're just different promoter regions for different genes in E. coli. And so you can see here various different genes given by their names here. Now, a few different regions. Now, transcription start site, you can see, is going to be TSS here. Transcription start site is given by these letters in code. So those are just going to be the first letters of the gene, or the first code in that coding gene. There's a region called the Prinbo box, which is about 10 or so base pairs upstream from the beginning of the gene. Of course, the gene would go that way, and this would be downstream. So upstream is before. And then there's even another region about 35 or so base pairs even further from the transcription site that all are involved in helping the RNA polymerase and the sigma factors to find out where they are supposed to bind. So these are what we call promoter regions. Okay, so even the simplest of organisms have a large amount of DNA. So RNA polymerase needs to know which is the template strand, where to transcribe, which is the first nucleotide to be transcribed, and so on. And so the DNA sequences essentially provide direction for the RNA polymerase. All right, so let's talk about initiation. So the part of transcription where RNA polymerase binds to the DNA, the strands are separated, and the first nucleotide binds to its complement is called chain initiation. Then it begins when RNA polymerase binds to the promoter and forms a closed complex. So let's talk about what a closed complex is. Well, there's two complexes. There's the open and there's closed complex. So the closed complex is the complex that initially forms between RNA polymerase and the DNA before transcription begins. And so that would be, here we've got RNA polymerase. You can see RNA polymerase and the sigma factor. So this would be the holoenzyme here. Sigma factor and RNA polymerase both contain complementary pairing to that promoter region. So that's going to help line them up in the right spot. And when they bind to the DNA, this is called a closed complex. Now, the open complex forms, the closed complex is what it's called before transcription. The open complex is what it's called during transcription. Now, there's a shift that happens. So the unwinding of the DNA doesn't give us an enzyme here that does this, but this unwinding of the DNA with the binding of these two guys already here is the open promoter complex. So then once the strands are separated, we have something called a transcription bubble, and it's about 17 base pairs or so long, and it moves down the DNA sequence to be transcribed. Now, RNA polymerase catalyzes the formation of these phosphodiester bonds as the ribonucleotides are coming in and being incorporated. Topoisomerases that we know about already from our topic on DNA replication. These relax the supercoils ahead of and behind this transcription bubble. You can see here what doesn't happen, okay? What doesn't happen is that RNA polymerase doesn't just run around the template strand following it in a big, following it around the helix. If that happened, the mRNA would get wound up around the DNA. So what actually happens is the topoisomerases unwind and wind ahead of and behind this transcription bubble so that the mRNA can come out and not be all tangled up, essentially. Okay. All right. So then, so this is the what we call chain elongation, and for chain elongation to actually even get started, there's a little bit that it has to overcome, right? We want the RNA polymerase and the sigma factor to recognize the promoter. So they come in, they bind to the promoter. But that binding means that they like that region. They want to stick there, right? And so it's hard to get them to come off of that. That's the difficulty in going from initiation to elongation. What this requires is that RNA polymerase kind of scrunches the DNA into itself, causing some torsional strain with the separated DNA strands, and then it launches itself forward. And so what we're looking at here is actually the ready to scrunch is the kind of caption on this image. It shows the open promoter complex of the bacterial RNA polymerase poised to begin the RNA synthesis. Arrows here are showing the direction of motion of the DNA segments as the first nucleotides are kind of being combined. And then again, when this thing is allowed to, let's see, what do the colors mean here? We've got the, in orange, we've got the active site of the polymerase. Sorry, the template is in orange in the active site of the polymerase, and the pink strand is the coding strand. And so what you can see here is that it's reading this strand. It's got this strand separated. They're kind of pulling on each other, and it uses this to get off of the promoter region. It's pretty neat. So this is the scrunching phenomenon. Okay, but once we can scrunch and we get chain initiation started, then everything that we just talked about happens, and then we have to figure out how does this, or your body has to figure out how to stop the initiation. We call this chain termination, and there's actually a couple of mechanisms. There's intrinsic termination. This is not dependent on a protein called Rho. There is Rho dependent and Rho independent. So intrinsic termination does not depend on the Rho protein and is controlled by what we call termination sites. So these are areas in the DNA that cause a termination of transcription by basically generating hairpin loops and zones, a zone of weak binding between the DNA and the RNA. And so what you can see here is that this happens with these inverted repeats. When this DNA gets separated from the two strands get separated, you can see here that one of the strands actually folds into this kind of loop here. Let's see. Sorry. The DNA transcribes into the mRNA these inverted repeats. And so this right here is the mRNA, not the DNA, not the separated DNA. This is the mRNA. And while this mRNA is kind of floating in the cytosol, being ready to be translated, wait, not be translated. Sorry. I'm thinking of something else. I'm thinking of termination in translation. This is termination in transcription. So let me just, everything that I said was mostly true. Let me just back up and be clear. The mRNA that's formed from these inverted repeats on the DNA causes the mRNA, as it's being transcribed, to fold into this sort of structure. Okay, and when this little structure forms, it creates zones of weak binding, and so this just causes the mRNA polymerase, the RNA polymerase to sort of dissociate from the DNA. It kind of these structures sort of get in the way and pull these things apart, and so this is what we call intrinsic termination. Now then there's Rho-dependent termination. Rho-dependent termination also causes sequences, or also has sequences that cause hairpin loops to form. So these hairpin loops are a common structure. The rho factor first attaches to to a recognition site on the mRNA and starts to move along it behind the mRNA polymerase. So then it's trailing up along the mRNA, chasing behind the RNA polymerase that's running along the template strand. Now, when the RNA polymerase pauses at this termination site, so the mRNA gets to a spot where there is a hairpin loop that's formed, the RNA stops a little bit, has to wait, rho catches up, and when rho catches up, sorry, that's my doorbell. When rho catches up, it unwinds the DNA RNA hybrid and actually makes it let go. And so releases the mRNA and also releases the RNA polymerase from the back end of that template strand. So rho dependent involves a rho factor. They both involve these secondary structures, these hairpin loops that end up forming. Again, it's unclear in the book exactly where, because they're not showing it in these pictures, so I wish I could give you a better idea of where these hairpin loops are forming. They must be forming right in this sort of, so if this is the termination site, and again, this is a termination site here, it's encoded in here to start forming a hairpin loop. And so I would imagine that once the RNA polymerase gets here and actually starts coding, that it's this part of the mRNA that's forming these hairpin loops and then possibly just getting in the way here and making everybody fall off from each other, at least in this type of termination. Now, the hairpin loop in the rho dependent causes this guy to kind of stall and get stuck, letting rho catch up. Okay. There's a couple of ways to control transcription in prokaryotes. There's alternative sigma factors, there's enhancers, there's operons, and there's something called transcription attenuation. We're going to talk a little bit about these. So alternative sigma factors. So viruses and bacteria can actually exert control over which genes are expressed by producing different sigma subunits that direct the RNA polymerases to different genes. And so a couple of examples are there's a virus that infects the bacteria Bacillus subtilis. There's a couple of sigma factors that come out in response to E. coli being heat shocked. Anyways, these different, you can see here, these different sigma factors are going to help guide the RNA polymerase to specific parts in the genes. So let's see if we can figure out what's happening here in this example I'm showing you guys. So in early transcription, there's a specificity factor, host sigma. So RNA polymerase and this host sigma factor come in and they transcribe these early genes. So we get some early proteins, including a protein called GP28. Now, GP28 is itself a sigma factor. So now GP28 has been made, and it's going to go and actually recruit DNA polymerase, or RNA polymerase, sorry, to now go and find these middle genes and to transcribe them. Those are going to include some more sigma factors that will go and get RNA polymerase to go and transcribe other genes. And so by turning on, or by transcribing the genes that code for sigma factors, you can actually get RNA polymerase to go to other specific parts of the genome and start, transcribing those. Now, RNA polymerase has affinity, obviously, for lots of different promoter regions. And so what you can do is, what your body does, and we saw that in the differences, if we jump back with some of these, is that some of these have higher and lower affinities for RNA polymerase. And so there are genes that are transcribed more often than others because of complementary base pairing. A way of increasing production of a certain gene would be to not go in here and recode it, but to encode for a sigma factor that actually will have some of the coding to come in here and help pick out a gene and help get it transcribed more often. So your body's got some really neat creative ways of modulating how much of a particular gene gets made or not made. Okay, enhancers. Enhancers are DNA sequences. Enhancers are DNA sequences that bind to transcription factor and increase the rate of transcription. So a transcription factor is a protein or another complex that binds to a DNA sequence and kind of alters the base level of transcription. So let's say that we have DNA, RNA polymerase running around transcribing a gene, just like we've been looking at. Well, let's say that we want that gene to be transcribed more often. Well, there might be certain transcription factors that actually help recruit RNA polymerase to those sites more often when they're present than if they're not present. And so transcription factors are going to be pretty useful in, again, modulating how much of a gene gets made. Now, enhancers are molecules that will bind to transcription factors. So just like, remember, transcription factors, like other proteins, can be modulated, or like enzymes, might be allosterically or so modulated. So an enhancer could bind to a transcription factor and now turn on that transcription factor so that it'll go and promote transcription of a gene. Or an enhancer, I guess we could call it an inhibitor, a molecule can go and bind to a transcription factor and actually turn it off, making it less likely to go and promote DNA polymerase. Okay, so certain, for example, certain E. coli genes include sequences upstream of the extended promoter region. And so here I'm showing you an extended promoter region, which includes not just our -10 and -35 regions, but all the way up to something called an up element, which extends up to the -60 upstream, and then into this FIS site. Now, these FIS sites are binding sites for a protein called FIS. Okay, in the E. coli, there are genes for ribosomal RNA production that have 3 upstream sites called FIS sites or FIS sites. And these FIS sites are the binding sites for the protein called FIS. And so you can see here, these would be transcription factors. These FIS proteins would come and bind here and help recruit the binding of RNA polymerase and sigma factors because they would be here and interactions between them would promote all of this. So enhancers can help to increase the transcription of a gene. And again, the example I'm showing you is just that specifically in a gene for the ribosomal RNA production. So if this is a gene that makes RNA, a ribosomal RNA, upstream we have these binding sites for the protein called FIS. And FIS is just a transcription factor. It's just a protein or a complex of proteins that bind to the DNA sequence and can increase the level of transcription. All right, and it goes without saying that if we can enhance the level of transcription, we can also inhibit it. Let's talk about operons for a little bit. Now, and then I'll get to this image. So an operon is kind of what we've been looking at. An operon contains a few things. It contains something called an operator, and we'll talk about what that is in a second. It contains a promoter, and it contains structural genes. Now, structural genes are the actual genes that we're interested in. They direct the synthesis of a protein. So the structural gene actually encodes amino acid sequences. The structural gene can be under the control of some regulatory genes. And this could be physically adjacent to the structural gene, but it could also be far away from the structural gene. An operon is not transcribed all the time. It can be regulated by induction. Induction is the triggering of the production of an enzyme by the presence of a specific inducer. So triggering by inducer. Now an inducer is a molecule that turns on the transcription of a gene. So an inducer can induce, or by induction, turn on a gene that would have normally not been transcribed. Again, under a it's a structural gene that's under control of a regulatory gene. Okay, so let's go into a little more detail. So I've got a picture here of a regulatory gene. This is an E. coli. This has see, what is the name of this? This is the beta lactose, the lac operon. This is the lac operon. Lac. All right, and we've got a few things here. We've got so a few different genes you can see here: lac A, lac Y, lac Z. Okay, so the structural gene here, beta galactosidase, an inducible protein coded for by lac Z. So lac Z here makes beta galactosidase. Now you can imagine that if you don't live on lactose, you probably don't need this gene. This is going to rip apart galactose, this is going to let you metabolize galactose. Galactose is the sugar that you get from lactose. So if you're not living on milk sugar and you have a source of glucose, then you're probably not going to need this gene. But if for some reason glucose goes away, you're going to need this gene. And so this gene's not always on. And so this beta galactosidase isn't always being made. So this is coded for by the structural gene lac Z. Now there's a structural gene lac Y that encodes lactose permease. And then there's a structural gene lac A. This encodes trans-acetylase. And the expression of these three structural genes is all controlled by the regulatory gene lac L. So this is the regulatory gene right here that controls the transcription of these structural genes. Now, a regulatory gene, again, directs the synthesis of a repressor protein. Now, repressor proteins bind to an operator gene. So the operator gene is right here. So the operator. Now, a repressor binds to the operator gene, blocking the transcription and eventual translation of the structural genes that are under control of that operator. So this operator right here, it's called an operator because it binds to a repressor. The repressor is coded for by the repressor gene, or the regulatory gene. So this regulatory gene gets transcribed. DNA polymerase or RNA polymerase comes in here, transcribes this gene. It makes this repressor monomer. In this case, 4 of those together form a tetramer that bind to the operator and prevent transcription of the rest of these structural genes. Okay, and this regulatory gene can actually be quite far from the operon. Okay, doesn't actually have to be part of this operon. Now, the repressor protein made by lac L forms a tetramer when it gets translated, and it binds to the operator portion of this operon. Now, the operator is the DNA element to which the repressor binds. I've already identified it for you guys right here. Now, when the repressor is bound to this operator, nothing can bind to the adjacent promoter, and so we don't get the laczine getting transcribed. So this must be the promoter site right here. This is the promoter region. So because of that regulatory gene there on the operator, we can't get DNA binding. So the operator and the promoter together form the control site. And the control site is basically a way of modulating the production of the protein. All right. And so you can see here specifically that when this lac operon is actually induced when E. coli has lactose and no glucose available for as a carbon source. And so if lactose is present, then it acts as an inducer. The inducer comes in here and it actually blocks this regulatory protein from being able to go and bind to the operator. And so then now RNA polymerase will recognize the promoter and it'll transcribe the genes. and it'll transcribe. Bacterial DNA is usually transcribed linearly like this, so it'll just make all of these together as one big mRNA, and then as the protein gets runs through these, it'll just kind of spit out these peptides. Something must cut them, but they kind of do it all in one go. All right, and yeah, so when glucose is present and lactose, the cell doesn't do any of this because the presence of glucose actually is what we would call a catabolite repression. Yeah. Okay. So your book actually does it does talk about how glucose is able to bind, how glucose is able to be detected by the cell, and it involves in the gene. So I think your book's got the repressor binding site, and then there's the start of a structural gene, and then it's got the RNA polymerase binding site. This is RNA pol, and then it has something called a cap binding site. And this cap binding site is the catabolite activator protein. And this cap protein will bind to the cap binding site when it complexes with something called cyclic AMP, right? And I think in the presence of glucose, this doesn't happen. So in the presence of glucose, we don't get this complex happening. When glucose isn't around, then the cyclic AMP forms the complex with the cap binding with the cap protein, and then they bind to the cap site, and this allows RNA polymerase to come in here and bind. Because without that cap and the CAMP complex, then this process wouldn't happen, and we wouldn't make lactose. So two things have to happen. Lactose has to be present as an inducer, and we have to have this complex forming, which only happens if glucose isn't around. All right, sorry about that. Hey, there it is. There's all the stuff that I was just talking about. Okay, let's talk about the different types of control mechanisms that we see in genes. So they may be inducible, or they may be repressible, and they may be positively or negatively controlled. So you can see here that this is induction, and so here we have a repressor that gets made by the DNA. That repressor will normally go in here and not have a gene working. So negatively, this repressor is turning the gene off, but an inducer can come in and actually inactivate the repressor. So it's induced to turn on. Same with induction, though, we can have the opposite happen. So here we have a regulatory gene that's an inactive inducer that's made. In this case, it would be an activator, but it hasn't been activated. So the gene can be transcribed without it. Nothing's blocking it. But this thing here, when it is co-induced, becomes active, turns on, comes in here, and the binding actually turns the gene on, where here, binding had turned the gene off, and it's unbinding that turns it on. So a little bit of the opposite, right? As far as repression goes, so both of these were induced to change. Now here's just a repression. So we've got this operon creating this repressor. Now this repressor can be co-repressed, and so in this case, where it deviates from here, is that this job is to repress, and it can repress unless it's turned off. This one's job is to repress, but it needs to be turned on so that it can repress. So both of these are repressing. One of them is induced to turn on, the other is induced to turn off. By off, I mean to repress. In this version, we've got some repression happening. So this is an active inducer that's being made by this DNA. Now, this would have the effect of going and turning on transcription, but the repression here happens with this inducer. This co-repressor comes in here and actually binds to the inducer, creating an inactive inducer. So the terms here, right? Repressors normally turn off genes. Inducers normally turn on genes. As far as repression goes, you can activate a repressor to turn off a gene, or you can activate a repressor to turn on a gene. So this is activating a repressor to turn a gene on. This is activating a repressor to turn a gene off. And then as far as inducing a gene to turn on, you can have an inactive inducer that gets activated to turn the gene on, or you can have an active inducer that has to be turned, I want to say turned on to be turned off, but that doesn't make any sense. This active inducer normally represses, and then an inactive inducer would cause this to come off and to be a positively, or what's the word? A turned on gene. Yeah, a lot going on with this graph here. So negative control means turning off, positive control is to turn on. Okay. What am I showing you here? This is the TRP operon in E. coli. I think what I wanted to show you here is just an example of a pretty complicated looking operon. Here you can see there's a couple of control sites. We've got a few different structural genes all lined up in a row. The mRNA here actually makes quite a few different proteins, but you can see here that they all can do different jobs. So like this protein here on its own is just half of the component of a fully functional protein, actually a fourth of it. We need 2 of these chains and 2 of these chains to actually make a full protein here. This guy's a protein on its own, 2 subunits near each other for tryptophan. So all of these are actually involved in the reaction of synthesizing tryptophan. So first couple of structural genes give you an enzyme that you need to do a reaction, and then actually to catalyze two steps in the reaction, and then another enzyme to give you some more steps in the reaction, and then a final enzyme to give you the 3rd step in the reaction. So again, just an example of an operon. All right, transcription attenuation. So a type of transcription control in which the transcription is controlled after it has begun via pausing and early release of incomplete RNA sequences. And there's a couple of different possible hairpin loops that can form. There's what we call the 1-2 pause structure. When this forms, it causes a premature termination of transcription. There's the 3-4 terminator structure. This again causes a premature release of the RNA transcript. And there's the 2-3 anti-terminator. This allows transcription to continue. And so you can see how those kind of look. Now, the way that this ultimately works, and I'll give you an example with tryptophan. So tryptophan is actually involved in its own sort of synthesis here. So let's see, because we just looked at the tryptophan operon here. So when you're making tryptophan, right? Let's talk about how this works here. So if there's a lot of tryptophan, if you already have lots of tryptophan, then you don't need to make tryptophan. Can we agree on that? If you already have a lot of it, when you have a lot of it, it's going to get incorporated almost instantly. When you're making your peptide and you get to the part on the code of the mRNA that says put a tryptophan here, if you have a tryptophan, you're going to be able to stick it in pretty nicely and pretty quickly. That means you're not going to have a slow ribosome. Your ribosome is going to be going pretty fast. When your ribosome goes fast, certain structures will form compared to others. And so you can see here, this transcription terminator will form. This structure right here will form. And since the ribosome will catch up to it, it'll basically initiate or de-initiate this elongation, and we'll end up with transcription termination. But if there is not a lot of tryptophan, if tryptophan isn't plentiful, then what ends up happening is the ribosome doesn't move as fast along the mRNA because it's waiting for tryptophan to show up. This allows for the ribosome to be in different places when these hairpin loops are forming, and an anti-terminator loop might form, which will have the effect of essentially keeping the ribosome from catching up to any of the other structures if they do form. Again, this keeps the process going. So depending on the speed of the ribosome and which of these hairpin loops actually forms, we get differences in the termination. So again, what we're talking about here are structures forming in the mRNA, okay, as RNA polymerase is transcribing it. As RNA polymerase is moving along the DNA, leaving behind a trail of mRNA, ribosomes like to jump on them and instantly be running along them, making proteins, trailing their protein chains out. So that's what's happening here. And depending on which of these structures forms and where the ribosomes are while this is forming, the body can keep this going, or it can prematurely end the transcription. Okay, let's talk a little bit about transcription in eukaryotes. So each of the RNA polymerases in eukaryotes transcribes a different set of genes and recognizes a different set of promoters. So there's RNA polymerase 1 that's found in the nucleolus and in synthesizing precursors of most ribosomal RNAs. There's RNA polymerase 2 found in the nucleoplasm and synthesizes mRNA precursors, sometimes called RNA polymerase B or RPB. There's RNA polymerase 3 found in the nucleoplasm and synthesizes tRNAs and precursors of the other 5S ribosomal RNA subunits. And so on. So this is different than the 5 different that we saw, RNA polymerases that we saw, or at least the 5 different subunits that we saw in prokaryotes. So different kinds of polymerases different kinds of polymerase subunits, at least, in eukaryotes. And a few different kinds of polymerases on their own. Now, these are all that I'm showing you here, all types of RPB or RNA polymerase 2 subunits, and you can see here that they some of these subunits are repeated in some of the other RNA polymerases. And then here's just a typical architecture of the yeast RNA polymerase 2 during transcription. So you can actually see the DNA, the helical structure, the RNA in red, kind of coming off to the side. Template strand is shown in blue, the coding strand in green. Okay. Now, some differences as far as in the polymerase 2 promoter, as far as upstream elements. So in transcription, a portion of the sequence is closer to the 3' end than the gene to be transcribed, where the DNA is read from 3' to 5', and where the RNA is formed from 5' to 3'. So upstream elements are anything that's closer to the 3' end of the gene being transcribed than the 5' end, okay? Where DNA is being read in this direction, right, to make the template. So anything that far is called upstream elements. There's something called the Tata box. This is about 25 base pairs upstream from the transcription start site. The Tata box contains a lot of TAs, a lot of T's and A's. Again, these have 2 hydrogen bonds versus 3, so this is going to make pulling the DNA apart here a lot easier. Not as much hydrogen bonds to overcome. There are initiator elements. These are loosely conserved sequences around the transcription start site. And then there's a downstream regulator. These are more rare than upstream regulators, but this is a regulator that's happening within the gene as opposed to upstream of the gene. All right, so we're going to need to initiate transcription. Now, any protein that's not itself a subunit of polymerase II is what we would call a transcription factor. And you've got a lot of transcription factors. Transcription will begin by forming a pre-initiation complex where RNA polymerase and general transcription factors bind to the DNA. As far as general transcription factors go, there's about 6 transcription factors that bind to DNA and initiate transcription. And so you can kind of see some of the steps here. So we're going to have some transcription factors recognizing the TATA box. We're going to have other transcription factors here helping to recruit RNA polymerase II. Pre-initiation complex forms when we've got all of those 6 transcription factors and the RNA polymerase, will form an open complex just like we did in prokaryotes when the DNA unwinds, and then elongation can start in a similar fashion to how it did with prokaryotes. Elongation is controlled by pause sites. This is where RNA polymerase will hesitate. It's also controlled by what we call abortion or premature termination. So this can happen if we need to. And again, this happens with those anti-termination or termination hairpin loops that can form. There's a couple of classes of elongation factors. I don't think I'm showing you a picture of that here. Let me go back here. A couple classes of elongation factors. So kind of coming down here. There are positive transcription elongation factors and negative transcription elongation factors. Because in terms of classes, we don't need to know anything about those. Then termination will begin by stopping RNA polymerase, and there's usually a sequence. The sequence for termination is pretty common, or pretty much the same in all of the different eukaryotic sequences. AAU, AAA is a sequence for termination. So when RNA polymerase gets to the end of the gene, it should find that and then terminate. Now, as far as regulation of gene expression goes, we have things called mediators. Now, mediators are these giant protein complexes that actually bridge the promoter regions of the DNA with general transcription factors. They bridge the promoter and the general transcription factors with remote regions that are either silencers or enhancers. And so a silencer is one that would turn the gene off, and an enhancer is one that would turn it on. Now, these silencers or enhancers are regulatory sequences that turn on or turn off transcription. Obviously, you can tell by the name. And DNA looping is what brings these enhancers or silencers into contact with the transcription factors and polymerase. So by having these enhancers present, these mediators and all these other proteins sort of bind. This can help to recruit RNA polymerase to these regions to begin gene expression. And same with this silencer protein here actually goes and causes this mediator to take a different conformation, right, than it would have in the other form. And so we don't get RNA polymerase and transcription factors binding. And so again, these silencers and enhancers can be on completely different parts of the DNA, nowhere near these genes that are being transcribed, but because of looping and bringing other chains or DNA pieces together, they're able to regulate each other like this. Let's talk about transcription in the nucleosomes. So the presence of nucleosomes, right, DNA being all wound up and packaged, actually represses transcription. And so activation of transcription depends on a few things. It depends on the interaction of RNA polymerase with the promoter and the transcription machinery, but it also depends on the relief of the repression that's caused by the chromatin structure. And so we need two forms. We need chromatin remodeling complexes. These mediate ATP-dependent conformational changes to the nucleosome that leads to transcription. So you can see here we've got some complexes. We've got some chromatin here, and then we've got these proteins, these complex proteins that come in here, and with ATP they're able to unwind the DNA a little bit, so we can get some transfer happening. Or we can do what's called remodeling, where different stretches of the DNA become exposed. So here we've got a stretch we can cause these octamers to slide over a little bit, revealing different parts of the DNA. There's also histone modifying enzymes. These make covalent modifications to the histone core octamers. And so there's many sites actually on the histones for modulation or modification, such as acetylation of lysines, the methylation of arginines, the methylation of lysines, phosphorylation of serines. And that's what I'm saying right here, and you can see all of these different regions. Now, methylations and phosphorylations are going to have the effect, and acetylations, of removing a lot of those positive charges on the histones. Positive charges, if you remember, are what made it bind so tightly to the DNA. DNA is negatively charged. And so by removing the positive charges, and by also adding negative charges like phosphorylation, we can make the DNA kind of repel the histones. One other thing that they talk about sometimes is the histone code, and that's what this is referring to, histone code. The histone code, so there's many factors that are involved in the switch between active transcription and inactive transcription, or inactive genes. Obviously transcription is active, and when it's not transcribing, it's inactive. Histone remodeling complexes are present, and so some nucleotides can get phosphorylated, some nucleotides can get, so the phosphorylated ones look like the little green lollipops here. Some histones can be acetylated. These are these little blue squiggly lines that you're seeing here, acetylated histones. Cytosines get unmethylated, these little white circles here. And so transcription factors in RNA polymerase will have access, will be able to have access to the DNA. And transcription is when transcription is not active, more nucleotides are phosphorylated, histones are deacetylated, and cytosines are methylated. And so we can see here that when a gene is turned off, these are the structural features we see. We see a lot of these methylations on the cytosines. We see a lot of these deacetylations on the histones. And when genes are on, we tend to see the opposite: acetylated histones, unmethylated cytosines, and so on. So by doing these modifications, these changes to the nucleotides or to the histones, we're able to, again, turn on and turn off regions of the DNA. So this is called the histone code. All right, real quickly, let's talk about RNA interference. We have these proteins called dicers. And what happens is we can get some DNA from a virus or some synthetic DNA, or it could be pre-microRNA that's been transcribed from a gene. And these go in and get diced up into the mRNA, the microRNA final form, or these small interfering RNAs that came from that virus. Now, through a process that involves a few different proteins, these RISC complexes and these agoproteins, we essentially use these RNAs to run interference. So we break them into these pieces that can basically be used to bind to any of those viral mRNAs that get made from this DNA, right? Or just other mRNAs that we don't want. We have lots of mRNAs in our body. We don't always want them to be transcribed. So a lot of the RNA in our bodies is running around just running interference, either perfectly binding to the mRNA produced by that DNA, because we basically had a little template of it. This perfectly binding to the mRNA is going to lead to no mRNA to translate. Of course, there's a complex with these proteins. I didn't talk about the AGO proteins. These AGO proteins are involved in, they're called Argonaut proteins, and they help to like chop stuff up. When the binding is perfect, the enzyme slicer cleaves the mRNA, so there's nothing to translate. When the binding is imperfect, it just ties up the hairpin loops. Actually, there's some hairpin loops that form, and the RISC RNA complex can't be translated. So in both senses, in both ways, it stops the production of proteins by interfering with the way that the RNA polymerase will interact with the mRNAs. So yeah, that's what what RNA interference is and what small interfering RNAs typically do. Now let's see. I wanted to talk about post-transcriptional RNA modifications also. tRNA, rRNA, and mRNA get modified after transcription to give rise to RNA's functional form. There's a few types of processing in prokaryotes that can differ greatly from that in our bodies. One of the main differences is the initial size of the RNA transcript is actually bigger than what we actually end up with, because we have a few things that we've done to it. We add a leader sequence at the 5' end. We have a trailing trailer sequence at the 3' end. We also have introns that get cut out. It kind of shortens it. There's a couple types of modifications: the trimming of the leader and trailer sequences, the addition of terminal sequences, modification of the structure of specific bases, particularly in the tRNA. So what you can see here is post-transcriptional modification of a tRNA precursor. So we've got a tRNA precursor here. The tRNA, of course, has a few extra bits on it. So the first thing that has to happen is that the exonuclease trims the 3' end, and it looks like it cuts off a UAA from somewhere. So this UAA gets cut off, so now it's missing. What's the next thing that happens? It gets a transferase enzyme puts on a CCA, so we get a new ending on here. If this tRNA is going to be, it looks like it's going to get cut in half. So an endonuclease cuts it right here, and we end up with an immature serine tRNA. And then we also end up with an immature proline tRNA in this example. And then we've got some 3' end trimming that happens, and a CA gets added to the end here. This is all so that it can recognize the correct amino acid. And then this one as well. This serine went this way and had some modifications as well. Yeah, so just touching up on this one more time. So the precursor of several tRNAs is often transcribed as one big long polynucleotide sequence. And then after trimming and base modification takes place, they can become mature tRNAs. A usual type of base modification is methylation and substitution of the sulfur for an oxygen. These are typical, again, typical base modifications for tRNAs. I don't think we saw. Did we see a sulfur anywhere on here? No. Okay, ribosomal RNA. Now, processing of ribosomal RNA is primarily a matter of methylation and trimming to the proper size. Prokaryotes have 3 RNAs in an intact ribosome. We have a few more than that. I think we also have 3, but different sedimentation coefficients. Not that that matters for us. Base modifications in both prokaryotes and eukaryotes are basically just accomplished by methylation. Now, this is what I'm showing you here is some of those base modifications here. So you can see this is a methylation of that oxygen, methylation on this oxygen. This is the 2' methyl ribosyl moiety. So this is what these guys have turned into, right? Typical base modification. Now, so we looked at tRNAs. tRNAs get modified in that they get cut into separate pre-MR or immature tRNAs, and then they get 3' additions to make them be able to attach to the right amino acid. As far as the bases, they get methylations, and this is, again, in ribosomal RNA. Messenger RNA is a little different. Messenger RNA, we also have some modification that happens when we make these. This includes a what we call a 5' end a capping of the 5' end, and so the transcription of this gene leads to the mRNA. Now, this mRNA transcript has some introns in it that need to get cut out, so we didn't talk about that. But there's some splicing that happens, and I'll show you that towards the end. And then we have the mature mRNA. Now, it gets a 5' cap to protect this side. It also gets a 3' polyadenylating. So lots of A's added to the end. And then, of course, splicing is going to happen. All right. So the 5' cap is a guanylate residue that's methylated at the N7 position. Not that that matters. Let's see. And of course, even on these, the 2' hydroxyl of the sugar is also methylated usually. The polyadenylate tail, this is usually 100 to 200 nucleotides long. It's added to the 3' end before the mRNA leaves the nucleus. And it protects the mRNA from nucleases and phosphatases. Now, I did mention that the eukaryotic genome does have exons that code for mRNA sequences, and then there are introns that kind of split up our genes that are not expressed in the final mRNA sequence, and these have to be spliced out. So let's talk about what splicing is. So splicing is the removal of these intervening sequences. It requires cleaving at the 5' and 3' ends of the introns and then joining those two ends together. Specific sequences actually make up the splice sites, and there's a branch site within the intron that has a conserved sequence as well. So you can see that the splice sites and the branch site, so conserved meaning it's usually the same in all the genes. Depends on small nuclear ribonucleoproteins, or SNRPs. RNPs, sorry. Small nuclear ribonucleoproteins. These mediate the process, and then when the exons are actually spliced together, we get a lariat form in the intron. This is the lariat right here, and the exons are spliced together. Okay. We don't need to go over the details of that reaction, but you can see here that it does involve some nucleophilic attacks. Okay. Now there's also alternative RNA splicing that happens. So genes, some gene expression can actually be controlled at the level of RNA splicing. So like 5% of the proteins produced in humans have isoforms that are based on alternative splicing. Isoforms are different forms of a protein, again, produced by alternative splicing. And there's possible reasons for this. So there's possible reasons for isoforms. There could be two forms of mRNA in the same cell, two different genes that made two different mRNAs that are similar to each other. Or they can come from, they can be from different tissues. Sometimes, lactase dehydrogenase, we see that it can be in the muscles, it can be in the heart, it can be in the brain, and have different isoforms. And so regulatory proteins can affect the recognition of splice sites and actually direct alternative splicing. And so you can kind of see that. Actually, I don't have an image of that for us. But by taking by splicing at different spots. So let me go back here. So alternative splicing might give us exon 1 and exon 2 and exon 3 together as a gene, but a different one might give us exon 2, exon 3, and exon 4 together as a gene, or maybe exon 1 and 3 and 4, or maybe exon 2 and 4, different things like that. Different splicing can lead to different isoforms. I didn't have an image of that, sorry. And lastly, let's talk about ribozymes. So ribozymes can catalyze their own self-splicing. They're involved in protein synthesis. There's actually two groups. There's the group 1 ribozymes. They require an external guanosine. And an example of one of those is the self-splicing in the pre-RNA of a little protist called tetrahyamina. And again, self-splicing of their own pre-RNA. And then there are group 2 ribozymes, and these display lariat mechanisms that we saw, like right here. So, and these don't require an external nucleotide, so meaning something coming in with a guanosine as opposed to the guanosine being already built into, the structure. So ribozymes, self-splicing enzymes, or self-splicing and self-catalyzing versions of these, and of course, we're talking about protein and RNA complexes throughout most of this anyways. But remember, a ribozyme has catalytic activity where normally we see proteins doing that. Okay, so I know that was a lot. There was a lot going on in this chapter. But let me know if you guys have any questions. And again, sorry about any of the confusion going through this chapter. It's been a while since I looked at some of this material in detail again. But yeah, let me know if you have any questions. Don't get caught up on the details. Just learn the big picture, right? It's the big picture that we're after here. And it's okay to learn it for prokaryotes, and just realize that with eukaryotes, it's just a little bit more complicated. All right, guys.